A new vaccine that could help fight Alzheimer's disease has been developed by scientists. The vaccine was tested on mice. It targets a protein linked to Alzheimer's and has successfully cleared harmful cells in mice with the disease. After getting the vaccine, the mice had fewer amyloid plaques in their brains, which are misfolded proteins that form in the spaces between nerve cells. The mice were also less anxious, had less brain inflammation, and their behavior and cognitive abilities improved. A previous study showed that people with Alzheimer's have too much of a certain protein called SAGP in their brain cells. The vaccine targets this protein, which is often too high in a type of brain cell called microglia. These cells play a vital role in the immune system of the brain. They clear away harmful plaques, but they can also cause brain inflammation that damages neurons and worsens cognitive decline in Alzheimer's patients. The vaccinated mice exhibited behavior similar to that of normal, healthy mice, displaying greater awareness of their surroundings. Now, the next step is to see if it works in humans. If it does, it could significantly slow down the progression of Alzheimer's or even prevent it from developing.
Scientists discovered a common fungus that can infect the mammalian brain and cause harmful amyloid plaques similar to those seen in Alzheimer's disease. Amyloid protein clumps, a hallmark of Alzheimer's, were previously thought to result from intrinsic brain stress or inflammation. However, in recent years, researchers have detected a fungus named Candida albicans in the brains of individuals with Alzheimer's and other neurodegenerative disorders. It raises the possibility that external infections can breach the brain, triggering an innate immune response. In one study, researchers injected Candida albicans directly into the brains of mice and found that the fungus triggered two neuroimmune mechanisms. One mechanism involved a fungal enzyme called SAPS, which increased the permeability of the blood-brain barrier, allowing fungal cells to enter the brain. The second mechanism is triggered by another fungal secretion, prompting microglia to target and remove the fungi. These immune pathways effectively clear Candida albicans infections in healthy mice brains, usually within 10 days. However, when researchers disrupted the microglia response in mice brains, Candida albicans infections persisted longer. This observation suggests that amyloid beta-like clumps serve as sentinels against pathogen invasion, but their improper clearance by microglia could be detrimental. Blood tests for Alzheimer's are now commercially available. Previously, Alzheimer's disease has been diagnosed through a combination of cognitive tests and biomarker tests. Cognitive assessments evaluate memory recall and thinking abilities, while biomarker tests analyze brain imaging scans or samples of cerebrospinal fluid. These tests identify the accumulation of biomarkers linked to Alzheimer's disease, such as amyloid beta plaques and tau protein tangles. However, these diagnostic methods often fail to detect early stage of Alzheimer's disease, which is necessary to stop the progression of the disease. The development of blood tests for Alzheimer's has advanced, thanks to the discovery that signs of the disease can be present in the blood up to 2 decades before symptoms appear. Two notable blood tests, named PRECIVITY-AD and QUEST, have emerged as front-runners in this field. These tests measure the ratio of 2-amyloid beta. A lower amyloid beta ratio is a significant sign of Alzheimer's disease and can be spotted in the early stages of the illness, even before noticeable dementia symptoms emerge. While these blood tests offer newfound hope, we still need to interpret their results carefully. They only detect one of the biomarkers for Alzheimer's and cannot identify other forms of dementia.